Welcome, everyone. Ready for another deep dive? Today, we're going on a fascinating journey into the world of gene expression.
Sounds exciting.
It is, really.
Yeah.
We're going to be unpacking how that DNA code actually turns into, well, the proteins and RNA molecules that basically make us who we are.
Stuff of life.
Exactly.
Oh, yeah.
And get this, we're exploring a whole chapter filled with mind-blowing theories, concepts, real-world examples, all that good stuff. So yeah, get ready to unravel how those genes of yours actually make things happen.
It's amazing how much information is packed into our DNA, isn't it?
I know, right?
And this deep dive, it's really going to give everyone a solid understanding of how that information gets translated into those molecules that make life happen.
Totally. We're going way beyond textbook definitions here. We're talking albino donkeys on some remote Italian island.
Yeah.
We're talking about the kind of heartbreaking connection between like a single genetic glitch and sudden death in young athletes.
Yeah, powerful stuff. These real-world cases, they really illustrate these concepts so vividly. They show how much impact even the tiniest changes in gene expression can have.
For sure. And speaking of history, our journey starts way back in 1902 with Archibald Garrod. This guy, this physician, he was so ahead of his time.
Oh, absolutely.
He was studying this really unusual condition, one where people's urine turned black.
Black urine. It's called alkaptonuria, and this was Garrod's clue. It helped him link faulty genes to specific diseases. He realized that the black urine, it was a buildup of a specific chemical, all because an enzyme that was supposed to break it down was missing.
Oh, wow. So like a broken link in the chain.
Exactly. His inborn errors of metabolism concept was, well, it was revolutionary. He basically proposed that genes control our entire biochemistry. They provide the instructions for making enzymes.
So without the right enzyme, things get backed up and you get these unusual, sometimes really harmful symptoms. It's amazing he figured that out back then.
It really was groundbreaking work. And his ideas, they paved the way for future discoveries, like Beetle and Tatum's work in the 1940s.
Oh, yeah, I remember them.
They used bread mold.
Bread mold.
Yeah, Neurospora crassa, to be precise. It really helped to solidify that connection between genes and enzymes.
Mold. Okay, you're going to have to break this one down for me.
Okay, so they exposed this bread mold to X-rays, which caused mutations in its genes. And these mutations, they created what we call nutritional mutants. These were strains of mold that couldn't survive unless specific nutrients were added to their growth medium.
So by seeing what nutrients the mutant mold needed to survive, they could figure out which enzyme was missing because of that mutation. That's pretty clever.
Exactly. It led them to their famous one gene, one enzyme hypothesis, which earned them a Nobel Prize.
Impressive.
Yeah. It essentially states that each gene gives instructions for making one specific enzyme.
But wait a minute. I thought genes made proteins. And there are tons of proteins out there that aren't enzymes, right? Like what about the keratin in our hair or hormones like insulin?
You're absolutely right. Not all proteins are enzymes. As scientists learn more about proteins, they realize that one gene, one enzyme idea, it needed a little refining.
So how they refine it?
The thinking evolved. It shifted to the one gene, one polypeptide hypothesis. A polypeptide is just a chain of amino acids. And proteins, they're made of one or more of these polypeptide chains. So the revised idea was that each gene actually codes for a specific polypeptide chain.
Okay, that makes sense. So genes, they're like the blueprints, polypeptides are the building blocks.
Right.
And then these polypeptides, they come together to form all these proteins. And those proteins have all these different jobs in the cell.
You got it. But the big question is, how do we actually get from a gene on a DNA molecule to a functional protein? That's where the really cool processes of transcription and translation come in.
Okay, so let's dive into those. First up, transcription. I know it has something to do with copying DNA instructions into RNA,
Yeah.
but can you walk us through like the step-by-step?
Right. Imagine DNA is a master cookbook, and RNA is like a copy of one of those recipes.
Okay, I like it.
In transcription, there's this enzyme called RNA polymerase. It binds to the DNA at a specific spot called the promoter. This basically signals the start of a gene.
So the promoter is like a bookmark telling RNA polymerase where to start reading the DNA sequence.
Precisely. Then, RNA polymerase, it unwinds the DNA double helix and starts to build an RNA strand based on that DNA template. It adds complementary RNA nucleotides one by one, and that creates what we call messenger RNA or mRNA.
mRNA. That's the one that carries the genetic instructions from the DNA in the nucleus out of the ribosomes, right?
You got it.
That's where the proteins are made.
Exactly. But in eukaryotic cells, like the ones we have, the RNA molecule goes through some extra processing before it can leave the nucleus. Think of it like editing a rough draft before it's ready to be published.
Oh, that's interesting. So what kind of editing are we talking about here?
Well, one important step is adding a cap to the beginning and a tail to the end of the RNA molecule. These help protect the mRNA and make sure it gets to the ribosome safe and sound.
Makes sense. And what about splicing? I've heard about that. That's where sections of the RNA molecule are actually cut out, and then the remaining pieces are joined back together, right?
You're right, splicing is crucial. It turns out eukaryotic genes contain these non-coding regions called introns. Those got to be removed. The remaining coding regions, called exons, those are spliced together, and that creates the final mature mRNA molecule.
So it's like editing a film. You cut out the unnecessary scenes, the introns, and you join the important parts, the exons, to make the final masterpiece.
Perfect analogy.
Yeah.
And here's where it gets even cooler. This splicing process, it can actually be different in different cell types.
Really?
Yeah, leading to slightly different versions of the same protein being made.
Wow. So you can have the same gene, but thanks to alternative splicing, different cells can make slightly different versions of the protein depending on what they need.
Exactly. It really highlights how complex and flexible gene expression really is.
That is amazing.
And speaking of complexity, let's move on to translation. That's where the mRNA molecule gets decoded to actually build a protein.
Okay, bring on the protein synthesis. So this happens at the ribosomes, right? What goes down there?
Ribosomes, think of them like little cellular factories,
Okay.
where amino acids are linked together to create a polypeptide chain, all based on those instructions in the mRNA.
So the mRNA is like the blueprint. The ribosome is like the construction site where the protein is built.
Exactly.
But how does the ribosome know which amino acids to use and in what order?
Well, that's where the genetic code comes in. It's this remarkable system where groups of 3 nucleotides on the mRNA, we call them codons, each specify a particular amino acid.
Hold on, so just 4 different nucleotides can code for 20 different amino acids. How's that even possible?
It's all about the combinations. With 3 nucleotides per codon, you have 64 possible codons, which is plenty to code for all 20 amino acids. Some amino acids are even coded by multiple codons.
So it's like a language with a limited alphabet, but tons of different words that can be made.
Yeah.
It's amazing how such a simple system can create so much diversity.
Absolutely. And this genetic code, it's practically universal. It's the same in organisms from bacteria to humans. It really shows how connected we are through evolution.
That is just mind-blowing.
Yeah.
So how does the ribosome actually match the right amino acid to each codon on the mRNA?
That's where transfer RNA or tRNA comes into play. These molecules, they act as translators. Each carries a specific amino acid and recognizes a particular codon on that mRNA.
So each tRNA has a kind of anticodon that matches up with the codon on the mRNA.
Exactly. The tRNA binds to its matching codon on the mRNA, bringing the right amino acid to the ribosome. And then the ribosome, it acts like a catalyst for forming a peptide bond between those amino acids, adding them to the growing polypeptide chain.
It's like this intricate dance with the mRNA playing the music, the ribosome as the choreographer, and the tRNAs bringing in the dancers, the amino acids.
Love the analogy.
So elegant.
And as that polypeptide chain grows, it starts to fold and twist, getting into its final 3D shape. And that's crucial for its function.
It really is amazing how all these components work together so seamlessly to build a protein from a simple DNA sequence. It's incredible. There's still one thing I'm wondering about. Well, we were talking about those albino donkeys and the athlete heart conditions earlier.
Right.
And both are caused by these tiny changes in the DNA sequences, right?
Uh-huh.
So how do these changes, these mutations, actually affect the protein structure and how it functions?
That's a great question. It brings us to the whole concept of mutations. These changes in the DNA sequence, they can have all sorts of effects depending on the kind of change it is and where it happens in the gene.
So it's not just about if a mutation happens, but also where it hits that DNA sequence.
Exactly. Take those albino donkeys, for example. Their lack of pigmentation, it's caused by a single nucleotide change in a gene called tyrosinase. This gene, it provides the instructions for making the enzyme tyrosinase. And tyrosinase, it's essential for making melanin, the pigment that gives color to our skin, hair, eyes, you name it.
So one tiny change in the DNA, like one single letter in the code, it leads to a non-functional enzyme, and boom, completely different appearance.
It's wild how these small changes at the molecular level can have such big effects, you know?
It really shows you how delicate this whole gene expression thing is. Now, with those athlete heart conditions, we see similar small changes in DNA with huge consequences. There's this condition called familial cardiomyopathy. It's a disease of the heart muscle, and it can be caused by single nucleotide mutations in genes that code for muscle proteins.
And these mutations can lead to heart failure, even sudden death in young, seemingly healthy athletes. It's scary. It makes you realize that genes aren't just about how we look, they're controlling how our organs work, our health, everything. Absolutely. So this brings us to the different ways these mutations can mess with genes and proteins. We can basically group them into substitutions, insertions, and deletions.
Okay, let's break those down. Substitutions, those sound pretty simple, like swapping one nucleotide for another, right?
Right. And the effect of a substitution, it all depends on where it happens and what change it makes to the protein. Some substitutions, they're silent. They don't actually change the amino acid sequence of the protein.
Wait, how's that possible? Doesn't each codon code for a specific amino acid?
They do, but remember, multiple codons can code for the same amino acid. It's like using different words but saying the same thing. The sentence changes, but the message is the same. So a silent mutation changes the DNA sequence, but not the amino acid sequence of the protein.
Interesting. So it's like a change that doesn't really do anything? What about substitutions that do change the amino acid sequence?
Those are called missense mutations. And the effect of a missense mutation, it depends on the specific amino acid change and where it is in the protein. Some are neutral, no big deal. Others can be bad, like changing the protein shape or how it interacts with other molecules.
So it's kind of like a typo in a recipe.
Mm-hmm.
Sometimes it doesn't matter. You swap one sugar for another. The cake's still good. But other times, one typo can ruin the whole thing.
Perfect analogy. And then we've got the worst kind of substitution, the nonsense mutation. This is when a substitution turns a codon that codes for an amino acid into a stop codon.
A stop codon. What's that do?
Think of them like periods at the end of a sentence. They tell the ribosome, hey, stop adding amino acids to this chain. So a nonsense mutation creates a premature stop codon, and you get this truncated, usually non-functional protein.
So it's like hitting a roadblock in the middle of a sentence, cutting the message short. The protein's incomplete, can't do its job.
Precisely. Now let's talk insertions and deletions. They're all about adding or removing one or more nucleotides from that DNA sequence.
Okay. Those sound kind of straightforwardly bad, like adding or taking away letters from the code.
Well, they can be problematic, especially when the number of nucleotides you insert or delete isn't a multiple of 3. Remember, codons are read in groups of 3. So if you add or delete 1 or 2 nucleotides, it shifts the whole reading frame of the gene.
Like shifting all the letters in a sentence by one space. Total gibberish.
Exactly. These are called frame shift mutations, and they usually end up with a totally different amino acid sequence after the mutation. Often, they lead to a premature stop codon, and you get a protein that doesn't work.
Wow, so even small insertions or deletions can totally mess up the protein.
They sure can.
Yeah.
Now, let's switch gears and talk about a really revolutionary technology that's changing how we can manipulate genes, CRISPR-Cas9.
CRISPR. I've heard so much about this gene editing tool. Seems like it could change everything, from treating diseases to maybe even enhancing human traits.
Yeah.
I gotta admit, I'm still a bit fuzzy on how it actually works. Can you break it down?
Of course. CRISPR-Cas9, it's based on this bacterial defense system that targets and destroys invading viral DNA. Scientists have basically hijacked this system to make a programmable gene editing tool. It lets us target and change specific DNA sequences with incredible accuracy.
So we're borrowing a tool from bacteria and using it on ourselves. Pretty clever.
It is. Basically, scientists create a guide RNA molecule that matches the DNA sequence they're targeting. This guide RNA then teams up with the Cas9 protein. It's an enzyme that acts like molecular scissors, cutting the DNA at that exact spot.
Like a guided missile, zeroing in on a specific gene and slicing it open.
Perfect analogy. Once the DNA is cut, the cell's natural repair mechanisms jump in. Scientists can then introduce a template DNA with the sequence they want. Basically, they trick the cell into repairing the break with the edited version.
Incredible. Like having a molecular scalpel, cutting and pasting genes with crazy accuracy.
Exactly. And this technology is already being used in labs all over the world. They're using it to study how genes work, develop new treatments for genetic diseases, even engineer crops with better traits.
It's amazing what we can do, but it also makes you wonder about the risks. We're talking about changing the very code of life. Are there, ethical concerns with CRISPR?
Definitely. And they're super important to think about. CRISPR has huge potential for good, but we also have to think about unintended consequences, and the possibility of misuse. We got to be careful, consider the ethical side, and make sure it's used responsibly.
For sure. Seems like with any powerful technology, there's always this balancing act between the good it can do and the need to be cautious and ethical.
Absolutely. But for now, let's get back to the basics of gene expression. We've been talking about genes as these regions of DNA that can be expressed, but what does expressed really mean?
Good question. I guess it's more than just the DNA sequence itself, right? There must be something that controls when and where a gene is actually turned on to make a protein.
You're right. A gene is more than just that coding sequence for a protein. It also has regulatory regions that control when and where it's expressed. Think of them like switches, turning genes on or off depending on different signals.
So it's not just about having the genes, it's about which ones are active. Like having a giant cookbook, but only choosing certain recipes based on what you feel like eating.
Love that. And this is where we get into the whole world of gene regulation. It's a complex system that lets cells fine-tune gene expression depending on tons of internal and external cues.
This sounds like a whole other deep dive on its own.
Yeah.
But can you give us like a quick overview of how gene regulation works? Who are the main players here?
Sure. One important level of control is at the transcription stage. Remember promoters, those sequences that tell RNA polymerase where to start transcribing?
Yeah.
Well, there are other regulatory sequences that can bind to proteins called transcription factors.
And those can either enhance or block transcription, right? So they're like volume knobs turning gene expression up or down.
Exactly. And there are all sorts of transcription factors responding to different signals. Some are activated by hormones, others by changes in nutrients, or even stress from the environment.
Wow, so it's like this whole network of signals and responses, all fine-tuning gene expression to make sure the cell is doing its thing, working right in whatever environment it's in. It's amazing to think that all this is happening at a molecular level.
It really is. And this is just one layer of gene regulation. There are also mechanisms that control how the mRNA is processed, how well it's translated, and even how long the protein sticks around in the cell.
It's like there's this whole other level of control beyond the basic gene expression stuff, like finding a secret room behind a bookshelf.
You got it. And this whole web of regulatory mechanisms, this is what allows cells to be so diverse and respond to their surroundings.
So we've covered a lot. We talked about the basics of gene expression, from that initial DNA blueprint to the whole dance of transcription and translation, all the way to manipulating genes with CRISPR and this incredible world of gene regulation. It's been an amazing journey. What are your final thoughts on why understanding gene expression is so important?
I think it really shows how genes aren't just like static blueprints, They're dynamic, always being regulated and fine-tuned to make this incredibly complex thing we call life happen.
It is incredible. Understanding gene expression is kind of like unlocking this fundamental language of life. It helps us see how our DNA shapes who we are, how our cells do their thing, even how diseases happen.
It really is.
It's like having the key to some secret code
Yeah.
that reveals the inner workings of our bodies and all the diversity of life on Earth. And with these powerful tools like CRISPR, it's like, wow, we're actually starting to figure out how to edit that code.
I know.
It opens up so many possibilities for like treating diseases and maybe even, enhancing human traits.
It's such an exciting time to be studying this stuff.
It is.
The field is always changing, new discoveries happening all the time. Who knows what's coming next?
I know, right? It's really inspiring to think about how this knowledge could like improve human health and well-being. And honestly, this deep dive has given me a whole new appreciation for just how complex and beautiful the processes are that make life possible.
It really is remarkable, isn't it?
Yeah.
I mean, from one single fertilized egg, you get this whole complex organism, trillions of cells working together in sync. And it's all thanks to this precise regulation of gene expression, making sure the right genes are turned on at the right time, in the right place.
It really makes you realize that we're not just a bunch of genes. We're like this intricate symphony of molecular processes, all orchestrated by our DNA.
Well said.
So we've reached the end of our deep dive into this amazing world of gene expression. We've covered the whole journey, from DNA to protein, looked at the details of mutations and gene regulation, even touched on the revolutionary potential of CRISPR.
We've covered a lot of ground today.
We have.
But as we've seen, there's always more to learn. The field is constantly moving forward. There are new discoveries all the time.
So if you're feeling inspired to learn more, keep going.
Yeah.
There are tons of resources out there to help you explore this amazing world even further.
And remember, even though all these processes we talked about might seem really complicated, they're all based on these basic principles that are honestly pretty easy to understand once you break them down.
Exactly, so don't be intimidated by the science. Just be curious, keep asking questions, and you'll be amazed at what you discover.
Until next time, keep learning and stay curious.